Okay, welcome back. So we are starting now the next presentation, which is by three persons, Serena Nik-Zainal, Jan Czarnecki, and Scott Shooter. So Serena and Tim, you can start.
Thank you very much, Nuria and EACR, for the opportunity to present some of our work today. I'm going to introduce the subject and then pass on to my team members, Jan Czarnecki and Scott Shooter. They are the brains behind the mutational signatures website Signal, so they will talk you through various aspects of mutational signatures and how to use the Signal website for both browsing signature data and performing your own analysis. So as a cell turns from being a normal cell into a cancer cell, as seen in the left side of the schematic, it doesn't just acquire a handful of driver mutations that we've heard a lot about in the first two presentations today. It also acquires a really large number of passenger mutations that arise because of various mutagenic processes that are going awry, in this schematic denoted as A to D. Some mutagenic processes may be perfectly benign, for example, process A, which are simply replication errors that arise every time a cell divides. We could expose the DNA in our cells to environmental mutagens like UV light or chemicals in tobacco smoke, as in process B, or we could acquire DNA repair defects, as in process C or D. Each of these mutational processes can leave a characteristic mutation pattern or imprint that we call a mutational signature on the genome. When you sequence a cancer genome, what you get is a final portrait, a composite of all those mutational signatures added together. However, in the past, it has been shown that if you had genome sequences from multiple tumors, it is possible to extract mutational signatures, i.e. identify processes A to D that have occurred in your tumors, and then to quantify them in each sample, i.e. determine the size of those arrows. I'm going to hand over to Jan Czarnecki now.
Peacock is a dataset of over 2,500 whole genome sequence tumor samples. Combining this dataset with other previously released breast tumor sample datasets, Andrea De Gasperi in our group was able to extract signatures from over 3,000 samples. A dataset of this size provided Andrea with the power to separate the dataset into 21 groups according to their organ of origin, and then extract organ-specific signatures from each group separately. This is something that wasn't possible with earlier, smaller datasets. This produced 192 organ-specific substitution signatures and 116 rearrangement signatures. These organ-specific signatures highlight inter-organ variability and are very useful when analyzing additional samples, which we'll come on to shortly. The organ-specific signatures were clustered together to produce reference signatures, which, in theory, are each the result of specific mutational processes. For instance, RefSig1 here is caused by the spontaneous deamination of methylcytosines, and as such, tends to correlate with age and isn't usually indicative of a problem. Here, in this plot, we can see the 96 different substitution types according to the original base, the base it is changed to, and the upstream and downstream bases. RefSig2, on the other hand, is attributed to the activity of the ApoBec family of cytosine deaminases, and RefSig3 is associated with a failure of homologous recombination repair. There are signatures associated with exogenous sources of damage also, such as RefSig7 here, which is found exclusively in skin tumors and is associated with UV light exposure. In total, we have found 38 substitution reference signatures, with the cause of many being unknown. The presence of specific mutational signatures in a sample provides clues about the history of the tumor, which may have treatment implications. For instance, tumors with a deficient homologous recombination repair pathway may be effectively treated with PARP1 inhibitors. By finding signatures associated with HR deficiency in a patient's tumor, such as RefSig3 here, but there are a number of others, they could potentially be provided the correct treatment earlier than they would otherwise. We have also investigated signatures experimentally in order to confirm the etiology of cancer signatures. For instance, this is RefSig MMR1, which is found in tumor samples with deficient mismatch repair. This is the signature found in MSH6 knockout cells. MSH6 is an integral part of the mismatch repair pathway. We can see clear similarities in the two signatures, and this provides further evidence that the cancer signature is the result of faulty mismatch repair. We have also extracted signatures from cells exposed to environmental mutagens, which can help confirm the cause of exogenous cancer signatures like REF67 on the previous slide. None of this data will be static. As more tumor samples become available, we will be able to refine our cancer signatures and introduce more gene knockouts and other analyses. All of this data is available on Signal at signal.mutationalsignatures.com. So let's have a look. Signal has been developed from the ground up using modern web technologies to create a user-friendly website. Signal has two main sections: Explore, where users can see the output of the team's research, and Analyze, where users can upload their own samples for signature analysis, which Scott will be talking about in just a moment. In Explore, we currently have the results of the 3 studies I mentioned previously. First, let's have a look at the cancer signatures. The main entry point into the dataset is this heat map. Along the top, we have the individual organs from which signatures were extracted from separately. And along the side, we have the reference signatures, which are the clusters of all of the organ-specific signatures. Each cell in the heat map displays the prevalence of each reference signature in specific organs. So we can see the reference signature 1 along the top here, which is caused by the spontaneous deamination of methylcytosines, is present in every single organ type. This is opposed to REF67 down on this row here, which is caused by or is associated with UV light, which is only found in skin cancer tumors. For each organ type, we can click on it, and we can see all of the organ-specific signatures that were extracted. In this case, breast tissue. We can see that there were 11 organ-specific signatures, and we can see the reference signatures that each organ-specific signature is associated with. We can click on each of these organ-specific signatures to see an interactive plot. We can see the associated reference signature, and we can also see the prevalence of this signature in all of the breast samples. We can drill down further and look at individual breast samples for any tissue type. We can see the profile of this particular sample. We can see the individual signatures that were extracted from this sample. If I go back to the heat map, we can come at this data from a slightly different angle. We can click on a reference signature, and here we can see the plot for this, for RefSig2 here, which is associated with the ApoBec proteins. We can see error bars here on each of the channels. This is because this is a cluster of organ-specific signatures. We can see here some annotations, which gives us a little bit more information. We can see the relevant cosmic signature for this reference signature, and we can see all of the organ-specific signatures which were clustered to form this reference signature. Can go back to the Explore page. We also have data for our experimental systems as well. So I'll click on the environmental mutagenesis page here. Here we can see all of the signatures that are extracted from cells exposed to certain environmental mutagens. I can search for a particular one here, in this case, simulated solar radiation, and we can see here that it's linked through to REF67 back in the cancer page, which is which it's very similar to. So that's just a very quick run through of the data we have. I'm now going to pass it over to my colleague, Scott Shooter, who will explain how you can analyze your own samples.
One moment, sorry. Thank you. So far, we've given you a brief overview of the methodology behind mutational signatures and shown you the breadth of mutational signature data that our team has generated. You've seen how you can explore both the sample data and the signature information in great detail on Signal. And I'm now going to talk in a little bit more detail about how we arrived at these signatures. And then I will demonstrate how you can use Signal to uncover mutational signatures in your own samples. Firstly, though, I wish to emphasize two key concepts that have already been mentioned, but it's very important to distinguish one from the other: signature extraction and signature fitting. Signature extraction is the process by which novel signatures are identified as patterns among a collection of sample catalogues. Signature extraction was used to generate all of the signatures that you've seen so far during this talk and in Jan's demo. Signature fitting, on the other hand, is the process by which a new sample, in this case a sample uploaded by a Signal user, is interrogated for the presence of one or more of the known signatures previously extracted. First, signature extraction. How do we extract novel signatures from a large collection of cancer samples? Well, we start with our N by 96 matrix. We have N samples, and each sample is described by its 96 channel substitution catalog. The aim of signature extraction is to explain our matrix on the right as a product of the two matrices on the left. The leftmost is a matrix of signatures and their 96 channels, and the other a matrix denoting the exposure or contribution of each of those signatures in each sample. So we have our equation, and in it there are two unknowns. To solve this equation, we use non-negative matrix factorization, or NMF. That is matrix factorization with the reasonable constraint that no cell in either matrix will take a negative value, as we cannot have a signature with a negative probability of mutating a particular base, and a sample cannot exhibit less than 0 contribution by any given signature. So we pick a random starting value for these two matrices, and using a KL divergence-based optimization algorithm, we iteratively change those matrices in search of a solution that minimizes our error. What we end up with is our two matrices that together approximate our samples, the most important of which being the matrix on the left representing the common patterns or signatures that we've extracted. So that's signature extraction. Signature fitting is a similar problem. We have essentially the same matrix of samples in the right, but in this case, it may just be a single sample, for example, one that a user has uploaded to Signal. And as before, we're trying to explain our sample matrix as a product of the two matrices on the left. The difference here is that we already have a matrix of signatures. These were found during extraction and are available on Signal, as Jan has shown. So the aim of fitting is to find a linear combination of these known signatures that is able to approximate our sample catalog. There is one issue with this, however, that Signal is uniquely positioned to fix. Fitting is a purely mathematical process, and the larger your pool of signatures, the more likely you are to find erroneous combinations that happen to approximate your sample purely by chance. So you need to limit your search space to just the signatures that are likely to be found in your samples. This is so that our solution can be optimally reflective of the underlying biology. We call these candidate signatures, and I'll show you in a few moments how Signal helps you with choosing them. So that's signature fitting, and that's what Signal provides, the means to perform signature fitting on your samples using the signatures that we've extracted. Furthermore, when you choose to perform signature fitting on your samples, Signal will run this algorithm 100 times over via a bootstrapping process in order to account for the possible noise that might be in your sample. So how do you do this fitting? Well, you begin by uploading your sample to Signal, and in just a few moments, you'll receive an array of results. You'll see your sample substitution catalog, you'll see the signature fitting results, and you'll see results for a number of other analyses that I'm not going to be covering today for lack of time, but that may be of interest to you. For example, evidence of any transcription strand bias among the mutations in your sample. We show you a mutation density plot that highlights regions of hypermutation, and we show you the similar samples in Signal's vast database. So I'll now show you a live demo of the analysis on the website. So if you want to upload your own samples for analysis, you come over to the Analyze section, and you're presented with a brief form to fill in. We allow you to upload your data in a number of different formats. If you have variant data, you would choose either VCF or CSV format. You can also upload catalogs directly if you have that instead of variant data. Click on the question mark just to see an example of each format. I'll choose the VCF, single sample. You can drag and drop your file here or choose the button to browse to the file on your machine. If it exists on a server, then you can paste the URL here. So I'll just pick an example file, choose the reference build. And here is where the parameters for signature fitting are configured. I previously mentioned that it's very important to select a limited subset of candidate signatures in order to minimize the likelihood of producing erroneous solutions by chance. To this end, Signal encourages you to select the tissue type of the cancer sample you're uploading. So I'll choose breast cancer, for example. Signal will then automatically pre-select just the signatures that were extracted from breast cancer samples specifically by our team. It leverages the power of the cancer-specific signatures that Jan talked about earlier on in order to produce the most accurate signature fit for your sample. You can also handpick signatures directly from a different number of categories if you click on the advanced options. But I'm going to leave that alone for now. Give the project a name and submit. Now, this will only take a few moments to analyze, but in the interest of time, I'm going to recall a previous result. All results will be generated and stored in the background anyway, so you're free to explore the site or even leave the site, and the results will still be there when you come back. So here's the result for this sample. First things first is the sample's 96 channel catalog that was generated from the variants. If we scroll down just beneath, we see the signatures that have been fitted. We see that Signal has found contributions from reference signatures 8, 3, 2, which is one of the APOBEC signatures Jan mentioned earlier, 18, and MMR1, one of the signatures indicative of potential deficiency of the mismatch repair pathway. Scrolling further down, we get a view of how accurately Signal was able to recreate the input sample catalog by combining the signatures assigned during the fit. In the background in grey, we have the observed sample catalog, and in the foreground, we have the catalog as recreated from the model. Cosine similarity in the corner here of 0.978 shows that the algorithm was able to do a pretty good job of reconstructing our sample. Regardless, there are still some mutations that went unaccounted for, and the difference between the observed and reconstructed profiles is isolated in what we call the residual plot below. There is another way of assessing the robustness of the fit. I mentioned earlier that Signal bootstraps the fitting process. So if I click on the advanced details of the results, I can assess how stable the solution is for each signature across all of the runs using this box plot. The median result for each signature is what is selected as its final result. The dashed green line represents a sparsity filter. So if a significant number of results for a given signature fall below that line, then that signature is excluded from the final results. It's a way of ensuring that the algorithm favors a solution for which it has a reasonable amount of evidence. I can also look at the correlations between the signatures down here. A highly negative correlation might indicate that the contribution of one signature is masking that of another similar signature, for example. Let's look at the two APOBEC signatures. So while in our results, RefSig2 falls above the threshold and RefSig13 below it, we can see that the two signatures have a slight negative correlation. This means that when one signature is more prevalent in a solution, the other tends to be less so. This might cause you to question somewhat the stability of these assignments, and while we do everything we can to present the most reliable results possible, it's important to be critical of any results you see, and to experiment as well. In this case, though, I feel fairly confident that at least one of the APOBEC signatures is present. On that note, I want to briefly show you one more feature of the fitting process. So suppose our sample has a signature that wasn't found by the algorithm because it wasn't selected as a candidate signature. Perhaps our sample has been exposed to additional mutational processes not found in the breast cancer samples during extraction. Just one example. To demonstrate such a situation, earlier I reran the sample with the APOBEC signatures removed from the candidate signatures. But we know these to be present, so we expect to see them. So with those now removed, if I look at those results and come down here, you can see that Signal's been able to identify the missing signature RefSig2, even though it wasn't selected as a candidate. And it does this based on the shape of the residual, which I showed you before down here, being a high match with RefSig2. And if you upload multiple samples at once, Signal might also suggest potentially novel patterns based on commonalities between your samples. So that's all I have time for right now. There are more results and features to examine, so I hope you'll hop onto the site and explore for yourself. Just want to thank our team in the MRC Cancer Unit, in particular Andrea De Gasperi, who laid a lot of the foundation for this work. Thanks for your attention, and please visit us on Twitter @SignalCambridge, where we'll be posting regular updates and new features.
Wow, it's an amazing tool. Congratulations to all the team. And we have a good number of questions waiting for you. So Daniel Brewer is the first one.
Hi there. I mean, it's a question you probably get quite often. I was just wondering if you could compare and contrast your approach for the reference signatures to Ludmil Alexandrov's, because they're on Cosmic and seem to be very popular in the community. So is your approach more robust, and should we really be using yours?
Yeah, thanks for the question, Daniel. Serena, could you take this question?
Sure. So in the original paper, so I think that's kind of less relevant to the website. The website sort of presents the information that we've produced in the paper. In the paper, you know, it's really we do a comparison of what we find to metastatic cancers. And we show that there is this organ-specific variability of the signatures, not in all signatures, but in quite a lot of them, and that we can recapitulate it in the metastatic cancer data set. So we work with exactly the same primary cancer data set that the Peacock and the original Nature paper that Ludmil has. You know, it's a little bit, I guess we're trying to put forward an alternative view, which is that there isn't necessarily a single signature that is the same across all tissues, and that actually that probably is a general mutational process, but that it has some slight variation from one tissue to another. So in terms of the actual reference signatures and cosmic signatures, there's a lot of similarity. A lot of it's very, very similar. Signature 2, ApoBex, signature 1 was all very similar. Signature 3 is a little bit different, so there are some differences, and it's very hard for us to know which one is right or which one is wrong. And I think we have to be a little bit careful that we're not trying to say our approach is better or our approach is more robust. But certainly, once you have the signatures, we have to ask the question, why are people using the signatures? And if people are using the signatures to make decisions about patient samples, then what you want to do is you want to try your best to make sure that the assignments to the signatures are as correct as possible. So if you do have a little bit of tissue variability, and there's a signature 3 that's more specific to breast or to ovary, then probably you would want to fit the signature that is more representative of your tissue to your samples. So I think that's how I think about it. But you know, at the end of the day, what you use is really dependent on the question that you have. Sorry, that was a very long answer.
Okay, thank you for that. And a bit more software-y type question to follow up. I mean, the website looks really great and is amazing, but many people would prefer to perform the analysis in R using something they're familiar with. So is there going to be an R package available, or can you use your signatures in one of the many tools that are already out there?
You can get the probability. Oh, sorry, Jan.
No. Finish what you were saying, Serena.
So you can get the mutation probabilities, the probabilities for the signatures, the tissue-specific signatures, as well as the reference signatures. You can just download that off our website, and then you can use that anyhow you want. Because all these different tools, you know, SIG profilers, somatic signatures, what they're trying to do is what the analyze component of this website does, which is the sound, right? So you can use any tool you want, but you can download the signatures from our website directly.
Great, thanks.
And just to, sorry, if you don't mind, I'll just add to that, is that Andrea and our team developed the signature tools lib R package, which essentially does what you say, and that's available open source on GitHub that you can download and use in R. And much of the code in that is used in the back end of Signal, but we more kind of developed Signal to kind of bridge the gap between people who are very computationally competent and people who are interested in the field, and the field is relevant to their work, but they may not necessarily have access or the skills to be able to use R packages. So the Signals kind of fills in that middle ground.
Great, thanks.
Thank you.
Okay, next question by Verena.
Okay, yeah, thank you. So I was interested whether you can also, whether there are also signatures available from the NCI cell line panel, just to make sure or to compare how predictive the the responses of NCI cell line investigations are for real cancer samples.
Shall I take that one, Jan? Sorry.
Yeah, go for it.
So we have not sequenced the NCI cell line. I don't know whether anyone else has. Yves Pommier has certainly spoken to me about it, but we haven't got access to any sequencing data to do signatures on that data set so far.
Okay, Natalie Conte.
Hi, thanks for this very useful tool. So I have a bit of a naive question to start with, which is, did you try to examine signature from primary versus metastasis from the same organ, and would you expect any differences?
So all of the data on Signal is, well, all of the cancer data is from primary tumors. We don't have any data from metastasis on there. There has been work in the team looking at metastatic tumors. But Serena, do you want to say, I don't know what is what would be ready to say?
Sure. So actually, in the De Gasperi paper, when we claimed that there was tissue-specific variability, we also demonstrated that tissue-specific variability was present in metastatic cancers. In addition to that, Nuria has published on signatures in metastatic cancers as well. It's the same cohort. So there has been work by others and by us on metastatic cancer signatures. However, it's not as extensive as the work on primary tumors. So primary tumors were worked on right at the start from 2010, 11, 12, and the Peacock cohort was primary cancer. So a lot of the stuff that you're seeing right now, a lot of these signatures are basically primary cancer signatures. So it's not a naive question, it's a very good question. And I think what's missing in the community right now is to look at metastatic signatures where we have removed all the primary mutations. So the metastatic cancer cohorts that both Nuria and we have looked at, those metastatic cancers, they didn't have a primary sequenced. So we couldn't remove all the primary mutations. So it was a mix of primary and metastatic signatures. So the specific analysis on just metastatic signatures has not been achieved so far as I'm aware. I could be wrong at the moment, but there might be people working on it. But so far, we haven't got anything like that. Are you aware of anything, Nuria?
No, it's not easy. You can analyze the mutations in the metastasis, but as Serena is saying, you will have the whole history of these cells, which include all the mutations that happened before the metastasis. So it's not easy at all.
Yeah.
You can play a bit with the timing of the mutations, but it's not very accurate. Yeah.
Yeah, you will need to have a longitudinal study that allows you to get the primary matched metastasis to remove the noise from the primary, right?
Exactly.
Thank you. I have another more practical question about using the tool, the sample tool, which is when you upload your samples. Do you have minimum number of samples? And if you want to upload variant data, are you limited by the resolution, meaning that you can't use targeted sequencing? You need to use minimum exomes? That's my question.
Yes. So first question, there isn't a minimum number of samples. So we're just fitting the signatures that we have in Signal to your particular sample. So you can just upload one sample if you want. We do set a maximum, but that's just to so that our servers aren't overloaded. So that there is a sort of there isn't a strict minimum number of variants, but because we're basically categorizing each of the substitutions into 96 different channels, there needs to be enough variants to be able to do that. So we do put up a warning on the website if you upload something which has a relatively small number of variants. I think it's 200 or less, and if it's 200 or less, its signature fitting likely isn't going to work very well. And so, saying that, with exome sequences, you can potentially, if there is a hypermutation phenotype, you can still potentially fit signatures to that. It's really down to the number of variants that are in the particular sample.
Thank you very much.
Okay, so the next question is by Simon Baker, but because he doesn't have a microphone, I will read it. Are there any plans to expand signal to analyze a wider flanking area around the mutation using ±2 bases, as was recently suggested by Ludmil Alexandrov in SIG Profiler Simulator?
Serena, do you have any thoughts about that?
We haven't automatically added that, and it's not in our sort of plans in our next release to go +2, -2. Currently on Signal, you can collapse your 96 channels into 6, for example, because sometimes you might not have very many mutations, and you may want to see what your mutations look like collapsed. The other thing is, because you have a lot of experimental data, and experimental data tends to be more sparse than cancer data, sometimes it's easier to look at things when they're collapsed. So right now, we have the ability to collapse mutations. I imagine it's not that difficult to expand, she said, offering the work to Scott again. But it wasn't in our immediate plans. I think the main reason for that is, although we can do it, you're still limited by power. So you know, when we first started doing this, even when we had in the Nature paper, Ludmil's Nature paper in 2013, we just didn't have the power, even though we had millions of mutations, to do it with +2 and -2. So that was point number one. And point number two is, what's the biological value of adding +2 and -2 for the loss of power? And so far, I think the only person who's kind of shown anything that's been very useful has been from Dmitry Gordinin's crew of people who kind of showed that there is this difference between ApoBac3A and ApoBac3B at +2, -2. But otherwise, so far, from a lot of people handling the data, we haven't really seen a huge amount of additional biology that we gain from the loss of power by introducing the +2, -2. So it hasn't been high on our agenda, but it's potentially something that is doable.
Next question is by an anonymous attendee, so I'll read it also. How many signatures are correct? Is signature extraction biased towards K higher than 1?
Oh dear, this is a very long question to answer. Nuria was already smiling when she read the question. This is it's a multiple solution problem. So it is a recurrent theme that it's very difficult to be absolutely certain how many signatures are in your cohort. It's true. But there is some confidence in that, some of the common signatures, the signature 1, the ApoBecs, the mismatch repair signatures, when they're there, and you will see them. You might miss 1 or 2 of the rarer, lower frequency signatures in a population. So if you have 100 samples and only 1 sample has a specific signature, then that might be missed if you've got the wrong number of K. But in general, the big common signatures, the ones that people kind of want to work with, they tend to be detectable, even if your K, your K is out by 1. So instead of, let's say your cohort actually has 6 signatures and you've chosen 7 or 5, in general, it doesn't make too much difference for the big common signatures. And actually in the De Gasperi paper, when we do the comparison between the primary signatures and the metastatic signatures, we actually show that. We actually show that even if you got the wrong number of signatures, you still see that tissue specificity is still very correlative between the primary and the metastatic cohort. So nobody knows exactly how to do this very, very well. But it's a good question. We're still trying to figure it out.
Next is Elena Grassi.
Okay, sorry, I had to unmute. Thank you for organizing this event. My question is about RefSig1. I was wondering if the slope of the correlation with age and the mutation in REFC1 is the same for all tissue or not. Because if that's the case, it would seem that it's not dependent of the number of mitosis, and this should imply that deamination occurs independently from mitosis. And since I don't know it, I was wondering about this point. Thank you.
So there was a paper maybe about 3 or 4 years ago now that looked at reference signature 1 across different tissues. And there is a very clear correlation with age, but that correlation is slightly different from one tissue to the next. So it gets called a clock-like signature. You know, whether that is a clock that's due to mitosis or whether that's a clock due to time, I think there's a little bit of an argument about that in the community. I mean, some people in the DNA repair community will argue that actually that clock cannot possibly be uniform, that the rate cannot just be uniform, and that as a cell turns from being a normal cell to being a cancer cell, things go awry, right? So you get replication stress, and actually when you've got a lot of single-stranded DNA flapping about, it will deaminate. So it's, yeah, I think when we do the maths, we sort of simplistically say, you know, it's correlated with age, but we sort of assume that it is, that rate is uniform throughout the evolution of the cancer. But I'm not sure that's 100% true. But generally, you know, to answer your question, the slope of the correlation is different from one tissue to another.
Okay, thank you very much.
Next, Saravana.
Thanks for the opportunity. I kind of mistyped the question, but it's two parts. One is like, how often have you encountered that you need to revise a signature when you interpret the data from whole genome sequencing compared to whole exome sequencing? And the second part is like, is contagious, which is like mutations that are found clustered within a chromosomal region, are they associated with any of the signatures? Thank you.
So all of the data or all of the cancer data is from the is whole genome sequenced, and similar to how what I said with the regarding the fitting before, you can do extraction using exome data, but you're then limiting your power, and you're limiting to just a small subset of the genome. And signatures is really designed to work across, the whole genome, including non-coding regions. Serena, can you answer about the contagious regions? Yeah.
Sure. So yes, that's a very insightful question. Chromothripsis is associated. Is it chromothripsis or contagious? Your question.
Contagious.
Yes. So contagious has been found to be associated with rearrangement breakpoints. But what's interesting is, in particular, it's associated with rearrangements that tend to be clustered, so focal rearrangements in specific parts of the genome. So for example, if you have a HER2 amplification or CCND1 amplification or a complex rearrangement or chromothripsis, then you're more likely to see contagious around there. Mm-hmm. There are some other rearrangement patterns which are more distributed through the genome, like you get in BRCA1 and BRCA2. You tend not to get contagious with those rearrangement breakpoints, but you do get it with things like chromothripsis and focal rearrangements.
Thank you.
One question by Piyush Kumar Mundra. You can ask the question yourself. And this will be the last question.
So basically my question is, like, there are multiple mutational signatures which are observed in each tumor genome. So how do we determine dominance of one signature over other in a given tumor genome?
Who's answering the question?
So you're basically trying to work out what signatures make up the overall profile that you see for a particular sample. So if you have lots of those, lots of samples, and the particular signatures are present at in different levels in different samples. You can effectively assign individual mutations to a particular signature in the extraction process. So when you look at so when each sample has a number of different signatures extracted from it, it comes with a number of mutations as well, which have been associated or which have been assigned to that particular signature. Does that answer your question?
It was actually in the analyzed part that Scott was showing, how he explained the process of fitting. And fitting just takes, you know, the signatures that you'd like to look for, and then tries to fit it to your sample to see how much of each signature is present in your sample. And so if your sample, for example, had a huge amount of ApoBec, then that will be the predominant pattern, and then that will be what will come out as the signature that has the greatest dominance. So if you show the profile of this one, Scott, there you go, that's the profile, that was your sample. And this is just all the mutations that have been counted. They've been added up for the 96 elements. This is what all the substitution mutations look like in your pattern. So now you know it's a breast cancer, and you're going to ask, which of the breast cancer signatures are present in my sample? And if you look further down that page, it's telling you the signatures that it's found are 8, 3, 30, 18, or whatever. And the biggest one actually was, here it says RefSig 8, but actually you can see signature 2 called at the bottom there. And actually, if you go back to the mutation profile, you can actually see it in the mutation profile there. You can see it there. Scott's pointing it out to you. So it actually tries to figure out how much of each of those signatures is present in your sample. So it will report to you which ones are the most dominant, but it'll report all of it to you, including the ones that are less dominant as well.
Yeah. Okay. Thank you.
Okay, Jan, Scott, Serena, thanks for a great presentation and discussion. And we have a 3 minutes break. We'll be back at 3:20. See you soon.